Myself, Dr. Pravita Kumar, Professor, Department of Chemistry, Shri Aurobindo College from University of Delhi. Learners, in previous class series of amino acids, we had discussed about the amino acids and we had learned there amino acids are the organic molecules or biomolecules which contain a basic group, that is the NH2 group, and an acidic group, carboxylic group in a same molecules. So we can say both the functional group, that is amino group and carboxylic group, present in a same molecule, in a single molecules called amino acids. And we had also learned their classifications of amino acids. In this class today we will discuss Zwitterion, that is the acid base characteristics of amino acids. Isoelectric points. On the basis of isoelectric points, how we can separate the amino acid from the proteins, or what is isoelectric points of amino acids. And on the basis of chirality, we can prepare a lot of drugs from the amino acids after the separation. And we will also discuss here how can we separate amino acids from the proteins or from the mixture. After separation, how do we identify the amino acids? So we will discuss the quantitative method of separation of amino acids. Learners, if we talk about the amino acids, there are two types of amino acids: essential amino acids and synthesis, which are obtained from the nutritions, or most of the plants we get these essential amino acids. And another type of amino acids, that is non-essential amino acids, and these non-essential amino acids are synthesized by our body. So these are the, on your screen you can see the amino acids which is present in human body. So there are two types, essential and non-essential. So non-essential, why they are non-essential? Because our body can synthesize these smaller biomolecules, these amino acids, while essential amino acids, why we call essential amino acids? Because we need to take these amino acids from our diet or from our nutritions, and these amino acids are take part in the protein synthesis. So the amino acids which takes part in the protein synthesis is called proteinogenic amino acids. So amino acids acting as a protein component also function in biosynthesis of protein in our body. So along with the protein synthesis, amino acids need for vital processes such as hormones and for the neurotransmitters. Learners, most of the amino acid, especially essentially amino acid, we get from the plants. So plants provide all the essential amino acids. Legumes and beans are great source of high quality of proteins. At least 20 to 25% of their protein is rich in amino acid, and the amino acid present in these green food or green plants, that is lysine. So legumes and beans are rich in amino acid, that is lysine. Also peas and beans contain 17 to 20% high quality of protein, while lupins and soya beans contain 30 to 45% of amino acids or proteins, because proteins are made up from the amino acids. If we talk about the structure, as I told you, amino acid are the molecules which contain amino group, which is basic in nature, and a carboxylic group which is in acidic in nature. So, you can see on your screen the carbon which is bonded to one end is amino group and other end is carboxylic group. So, unique characteristics of these amino acids are presence of free amino groups in alpha carbon atom and also a free carboxyl group present in amino acids. Nearly all amino acids exist as a Zwitterion, and Zwitterion provides strong. Why is amino? What is the role of that Zwitterion, and why all amino acids present in the form of Zwitterion? What is the reason? Because Zwitterion provides strong hydration through electrostatic interaction with water molecules and provide a physical and energetic barrier against the protein absorption. And also, because there are Zwitterion, amino acid have many physical properties, and that properties we can associate it with the salt, because positive charge and negative charge, both the charges are present in the amino acid. So, we can, the properties of these amino acid we can associate with the salt, like we can form the crystals from the amino acids, and also they have high melting points, or they are soluble in water, and they are not soluble in hydrocarbon solvents. On your screen you are looking amino acids, and as I told you, the amino group which is present in alpha carbon atom, that is basic in nature. Also, carboxylic group which is present on chiral carbon atom, which is acidic in nature. So, both the acidic and basic forms we can find out in the amino acids. But how can we decide the amino acid is in basic in nature and acidic in nature? So, learners, it depends upon the pH of solution in which amino acid is dissolved, like when internal transfer of the hydrogen, because there is a COH group on the one end and NH2 group on the another end. So, when internal hydrogen transfers from that carboxylic group to amino group, it leave an ion with both negative and positive charge. So, due to internal transfer, or we can say due to internal transfer of a proton which is present on the carboxylic group, also NH2 having also 2H. So, from due to that transfer of the proton, transfer of the hydrogen, it leaves amino acids as a dipolar acid and have both negative and positive charge. So, when it's very important, learners, you have to understand how the internal transfer from one carboxylic groups to NH2 group or NH2 group to carboxylic group takes place. For acid basic character of amino acid, you have to understand how the internal transfer takes place. So, as I told you, because there is a COH group, when hydrogen is transferred from the COH group, we call this deprotonation. So, transfer of hydrogen is called deprotonation. Similarly, NH2, protonation from NH2 also we can deprotonate or protonation. So, protonation separate charges in the solution on one compound, that is the amino acids. That's why due to internal protonation and deprotonations, the amino acid exists as a Zwitterion or dipolar ion, because on your screen you are looking here the carboxylic and having a negative charge while the nitrogen having a positively charged, right. So, due to that internal migration of, or it in a scientific way or in a chemical way we can say deprotonation and protonation. The amino acid exists as a Zwitter ion, and this is very important characteristics of amino acids, and due to that characteristics it shows crystalline properties due to Zwitter, formation of Zwitter ion. And as you have seen the previous slide, Zwitter ion is a diprotic. What is a diprotic? As you can understand by the term protic. Protic is associated with the hydrogen atom or a proton, because in during the formation of Zwitter ion, protonation or deprotonation, transfer or acceptance of protons takes place. That's why this is a diprotic in nature. Also, two groups which can donate or accept a proton, right? So, because that carboxylic acid which can donate proton or can accept a proton. Similarly, amino group can donate a proton or can accept a proton. So, that is why this is that Zwitter ion is diprotic in nature. And when Zwitter ion exists in a solution, we called it a neutral ion, because learners, there is a equal charge, equal positive charge and equal negative charge. Now, what is the protonation? You have to understand how the protonation takes place. Protonation means adding of a proton, and always you know what is the proton? Proton are the hydrogen which is electron deficient. So, always we have to represent proton as a H plus. So, adding of a proton when the concentration of, it means that what it means that when the proton is added to the molecule, when concentration of proton is more in the solution, that we call protonation. So, that's why the proton is added to the group. And when the concentration of protons is maximum in the solution, as I told you, it depends upon the pH. When the solution pH of solution is decreases, right? When we add more acids, what happen? The pH is decreases, and as the pH is decreases, the concentration of H is increases, and if the concentration of proton is increases, protonation takes place. And in a short form for your memory, I have to represent it here PDP, that is pH decreases, protonation takes place. So, when in a solution, when pH is decreases, protonation always takes place. Similarly, what is deprotonation? How the deprotonation takes place? Deprotonation means removal of a proton. And when the removal of a proton takes place, learners, when the pH of solution is increases, right? What does it means? It means that when the pH of solution is increasing, the concentration of proton is decreases, right? And when the concentration of proton is decreases, removal of a proton from that groups takes place, and this is called deprotonation. And I have represented here for your memorization, I have represented here PID, that is pH increases, deprotonation takes place. PID, P is pH, I increases, D deprotonation. So, pH when increases, deprotonation takes place. Now, you can see here that is a carboxylate ion. How that carboxylate ion accept a proton? When the pH is low, as I told you in the previous slide, when the pH is low, the concentration of proton is maximum, and that carboxylate ion accept a proton and protonation takes place. Similarly, in case of NH2, it is a deprotonation, and vice versa. We can understand over here COH, deprotonation is a removal of a proton. Deprotonation from the carboxylic group when pH is increases, and similarly deprotonation from the amino group when the pH is increases. So, due to depending upon the pH of the solution, that amino acid can be exist as a cationic form and anionic form in a solution. But learners, there is a middle point of the reaction when the reaction will be in equilibrium. And at what you will see here, effect of pH. What is the effect of pH takes place? If solution of amino acid is prepared by low pH, right? So what happened? The NH2 group which is present in the amino acid accept a proton due to presence of H proton ion, H plus ion in the acidic medium. So you can see here how the Zwitterion behave over here. In this structure, the oxygen is negative, nitrogen is positive. What it means that from the carboxylic part deprotonation takes place, and for the amino group protonation takes place. Now it is a Zwitterion. So after adding the acid, what happened? Learner, understand over here, it is a Zwitterion. So as I told you, Zwitterion is a neutral ion because there is a no net charge over here, because one negative charge, one positive charge, right? So what happened when we add acid into the solution? So when we add acid into the solution, it decreases the pH. When pH is decreases, as I told you, the concentration of proton is increases. So you can see when the concentration of a proton is increases, the Zwitterion no longer Zwitterion. You can see here because there is over here and one positive charge. So when the pH is increases, again carboxylate ion accept a proton, and in that case that Zwitterion is no longer dipolar ion, that is also having only one positive charge. Similarly, what happened when we add a base into the solution of Zwitterion or amino acid? So learner, you can see on your screen, when we add alkali or base, what happened? The OH, after adding the OH, the concentration of proton is decreases. It means that the proton from the amino group, ammonium ion, is deprotonated, and during that deprotonation, the positive charge is its positive charge. So again, in that case, the Zwitterion is no longer act as a Zwitterion. So shifting of, that is the pH, from one extreme to other ions in acidic solution or after adding of the alkali, what happened? The two acidic hydrogen, one in COH group, that is the carboxylic group, and one in NH3 group, that is in amino group. So when COH is greater than ammonium ion, it means the solution is a more acidic, and it remove, and after removing that we get a Zwitterion again. So here you can see here, this is the ammonium which is in cationic form having a positive charge. So if we get acid, we add acid over here into the solution, what happen again? Protonation takes place, and COH is more acidic in that case, and when we add light amount of alkali, amino acid is no longer, and as we get a Zwitterion again. And Zwitterion on your screen, you can say here both the positive and negative charge. So if we add more hydroxide ion, H has started remove from that ammonium ion, because the NH2 is a stronger base, it picks up hydrogen ion first and back to Zwitterion again. So that is why the amino acid always present as a Zwitterion, as a diprotic ion into the solution. So this is the example of glycine. So on your screen you are looking here neutral glycine. What is the neutral glycine? There is no charge. So COH and NH2 group can exist more acidic form on basic form depending upon the solution. So we can see here, as I explained in the previous slides, you can understand here there is a single step reaction how glycine exists as a dipolar ion or Zwitter ion. So in the first part you can see here that glycine exists as a cationic form. Why this is a cationic form? Because as I told you, when increases the pH, concentration of H is increases, and as the concentration of H is increasing, the NH2 group which is present in amino acid accept a proton. As this amino acid accept a proton, NH2 accept a proton, it becomes NH3 plus. So this is the cationic form, and this cationic form of amino acid, this is the glycine, predominant in strongly acidic solution. So always remember, the cationic form always predominant when the pH of solution is 0 or pH of solution is 0 to 2, 3, 4. Similarly, when in the last end you can see anionic form. So when anionic form predominates, anionic form predominates when there is a strong OH is over here. So strongly pH is over here. Here pH is 14. So as the pH is increases, the concentration of H is decreases, and as the concentration of H is decreases, deprotonation takes place from the NH2 group, and in between them there is a dipolar ion or Zwitter ion. So between the cationic and anionic form we can found a stable form of diprotic amino acid which is called dipolar ion or Zwitter ion, which have one positive charge and negative charge. So predominant form of amino acid in solution depend upon the pH of solution and the nature of amino acids. Also learners, side chain of amino acid, because the side chain which is R or alkyl group are different in all amino acids, that can be basic in nature, that can be ionizable, or that side chain can be non-ionizable. So it's very important, you have always remember, it is a representation of Zwitter ion in a scientific way is when the solution is acidic, it means that the pH of solution is increases, we get a cationic form, and in cationic form protonation takes place. Why this is protonation takes place? That NH2 is accept a proton and we get a NH3 ion. Similarly, when the strongly basic solution, anionic forms predominates, and in anionic form deprotonation from the carboxylic group takes place, that is the removal of protons takes place, and in middle of that is we get a dipolar ion. And learners, the pH at which we get Zwitter ion or dipolar ion is called isoelectric points, and at that isoelectric points the concentration of amino acid is a maximum. So on your screen you are looking again amino acid which is present in the both form, cationic form and anionic form, and in between of them there is a Zwitter ion. So at pH 7 there is Zwitter ion, and this pH is called isoelectric points. Here at that the carboxylic group of amino acid have pKa value is 2, it means the protonation of amino acids and that amino acid have pKa value near the 9. So these are the different amino acids, and they have a different pKa values for carboxylic group and pKa values for the amino groups. Along with that there is a isoelectric point pKa value for the side chains, because most of the amino acid except the glycine they are having different types of side chains. So both the group NH2 and COH in acidic solution we get a pH 0. So at pH 7 the pH of solution is greater than the pKa value of carboxylic groups, but that is less than the pKa value of the ammonium group. So, if carboxylic group will be in its basic form and NH2 group will be in acidic form. So, as here in strongly basic medium, the pH is nearly 11. So, both the group will be in a basic form, NH2 group and COH group will be in the basic forms. So, you have to important point, you have to always learn the amino acid can never exist as an uncharged compound regardless of the pH of the solution. And why does amino acid shows the Zwitterion formation? Because in aqueous solution amino acids are more stable in Zwitterion form. So, here in this part we had learned about the acid basic properties of amino acid, how these properties are depend upon the pH of solution, and if we increases the pH of amino acid solution in which we have taken the amino acid, the protonation takes place, concentration of H takes place, concentration will be more and deprotonation takes place. So, depending upon the pH of solution we get a Zwitterion, and Zwitterion is a form of amino acid where both the charge positive and negative charge present in a same molecules. Thank you.